My name is Mark Cowley. I'm a researcher in the pancreatic cancer group at Garvan. We use cancer genome sequencing to learn about carcinogenesis and also to discover molecular features of cancer that we can hopefully target with specific drugs. Essentially, through our work in sequencing these cancer genomes, we've developed approaches, developed software to enable this. But really, it's in the domain expertise of the the bioinformatician to actually run this software. So really, what this project has done is take some of the software and the approaches that we've developed, turn them into Galaxy modules to run in this Galaxy web-based system, which really allows then cancer researchers to do this type of analysis themselves. I think the big thing there is just enabling a different type of researcher to have access to this data. This whole project has really enabled far more people to analyse this data, and as the data gets more and more, it should be accessible to a whole range of people to analyse. We're really laying the foundation for personalised medicine here. I mean, we're still probably a long way off from having an automated pipeline that pops a drug out the other end, but through the tools that have been developed in this project, you can go from raw sequencing data coming off the machine all the way through to a list of mutations that's characterised by the impact on the genes that those mutations hit. And I think a talented molecular oncologist can take that type of information and link that to therapies. And at the moment, that's about the point where we're at. Sharing workflows is just incredibly valuable. I think you can get the bioinformaticians, the technical team to actually do the first version of the pipeline and choose the pieces in the pipeline and the default properties, the configuration of those, and then allow the researchers in the lab to just start using those. And then as they get more and more comfortable with using those, they can start editing some of the parameters, and then if they get really comfortable, they can make their own pipelines from scratch. I think the pipelining engine is actually really going to lower the barrier of entry to using these techniques. I think it's extremely important because we're trying to enable reproducible research. So we know that the software is rapidly evolving, but especially if we're trying to apply this in the clinic, you're going to need to be able to go back to old data with old versions of algorithms and reproduce those results identically. And then you can try and compare those to the more modern algorithms and see how it's changed. But I think being able to reproduce what you've done in a period of time, especially when patients are involved, is absolutely vital.